Good evening, this is Mr. Wintermelon. So in the previous lesson, we have learned the structure of DNA and the simplified version of how DNA is the genetic code that is used to synthesize your polypeptide chain. In this lesson, we will learn about how we can transfer the DNA of one organism to another. So by the end of this lesson, you should be able to explain that gene may be transferred between cells. Then briefly explain how a gene that controls the production of human insulin can be inserted into bacterial DNA to produce human insulin. And lastly, to discuss the social and ethical implication of genetic engineering with reference of a named example. So what is genetic engineering? Genetic engineering is the transfer of genes from one organism to another of be it the same or different species. The transferred gene must be able to express itself in the recipient organism. So you cannot just anyhow suka-suka like transfer one gene then say, oh yay, genetic engineering done. Cannot. Express down here means the gene is working. The gene is being used to produce the new protein. So the organism will be known as a transgenic organism. So take for example, you have a banana that have a disease-resistant gene. So you can transfer this gene to another gigantic banana. So now the gigantic banana is now both bigger and disease-resistant. So now the gigantic banana have the gene to make the banana bigger as well as a disease-resistant gene. So in the end, you will get a gigantic banana that doesn't get disease. Wow, win-win. So this is the rough idea of genetic engineering. So what you need in genetic engineering? So in this slide, it is just a list of new words that you have to learn. The first thing you need is a DNA containing the gene that you want. For example, insulin. Okay, gene is nothing new, but of course you need it. If not, you got nothing to transfer. Then you need an enzyme known as restriction enzyme. It is like a scissors to cut the DNA. So you need to cut the gene out using restriction enzyme. Then you also need this enzyme known as polymerase to make many copies of this gene which you want. So this enzyme called polymerase, not difficult to remember the name because poly means a lot and its function is to make a lot of the gene. Then you will also need a plasmid. Plasmid is actually the DNA inside bacteria. So the DNA inside bacteria isn't helical in shape. It is circular in shape, unlike our human. So you will use the same restriction enzyme to cut the plasmid. Then you will need glue to glue the gene into the plasmid. The glue here is the enzyme known as ligase. So once you have glued the gene into the plasmid will be known as a recombinant plasmid. And you will have to insert this recombinant plasmid into a vector. Most of the time is either a bacteria or a virus. And if this is successful, the gene will be expressing itself, producing the protein, and we can harvest the protein. So now let us watch a video of the whole process. What you will need is the DNA, which contains the gene that will express the desired protein, circular plasmid from the bacteria, our molecular scissors, the restriction enzyme. We will need two different restriction enzymes for this. You will also need DNA ligase to glue the gene onto the plasmid, which I will use my hand to represent it.
So the first step is to use the restriction enzyme to cut out the gene from the DNA. Once we manage to cut out the gene, we will use polymerase to make many copies of the gene so that it will increase the chance of it being introduced into the plasmid. Once we have done that, we will use the same restriction enzyme to cut the plasmid. Once the plasmid is cut, we can use the DNA ligase to glue the desired gene onto the plasmid. So once we have gotten our recombinant plasmid, what we can do is to transform it into a vector. So this vector can be bacteria or viruses. If it's a bacteria, we can grow the bacteria in fermenter, and the bacteria can express a lot of this protein which we can later harvest. If it's a virus, the virus can introduce the plasmid into a host plant. For example, if we want the plant to be resistant to some disease, and this gene produces the protein that have the disease resistant effect, then the virus can introduce this plasmid into the plant. So this is how genetic engineering is done.
So to put what we have learned in words, first we use the restriction enzyme to cut out the insulin gene from our DNA fragment. Then using polymerase, we increase the number of copies of our insulin gene so that it is easier for us to insert them into the plasmid. Using the same restriction enzyme, which we use to cut our insulin gene out, we use it to cut the plasmid. So now that the plasmid is cut, we can use DNA ligase to seal the insulin gene into the plasmid. So once the insulin gene is added to the plasmid, it is known as a recombinant plasmid. Then we have to insert this plasmid into our bacterium. So by applying heat and electrical shock, we can transform this recombinant plasmid into our bacterium. So now the bacteria can produce the protein insulin. What we can do is we can grow the bacteria in fermenter. Fermenter are places whereby it has the optimum condition for the growth of bacteria. That means bacteria will grow or multiply very quickly. So more bacteria will produce more insulin. So we will get a high concentration of insulin in the end. And in the case of other protein, whereby we want to insert the protein directly into the host cell, we can introduce the bacteria or the virus to the host cell. Then the bacterium or the virus can insert the plasmid into the host cell because they are actually quite good in it. But of course, this is not exactly inside your syllabus. That's why I am not highlighting this in red. So in summary, we use restriction enzyme to cut both the DNA and plasmid. Then we use ligase to stick or to seal the desired gene into the plasmid. Then we perform transformation of the recombinant plasmid into the host. Depending on the application of the protein, For example, if the protein is something that we want to harvest and let's say give to human, we can grow the bacterium in fermenter and harvest the protein. In the case whereby we want the organism to produce the protein itself, we can perform transfection of organism with bacteria or virus. Then gene will be inserted into the organism chromosome. Because bacteria and viruses, they actually have the tool to do that. Now, let us discuss about the benefits of genetic engineering. There are a few things to remember for the next few slides. So I am going to tell you the easy way of remembering. So when we talk about benefits of genetic engineering, just remember this material. What is this material? This is a PVC pipe, which we use a lot in construction or engineering. So what are the benefits? We look at P. P stands for pollution. Of course, not more pollution, but reduction in pollution of the environment. Why? Because we are using less pesticide. Why are we using less pesticide? We have to look at V. V for variety. Because the crops are of better variety. So those crops are genetic modified food and they are resistant to drought. They also have pest resistance. For example, the Bt genes allow production of toxin that kills insect. And because of this, we do not have to use pesticide which will pollute the environment. And better variety of crops also have higher nutrients. For example, the golden rice. So the rice grain have high vitamin A content. So Bt genes as well as golden rice are example of better crops variety. So these two examples, why do we genetically modify our food? So that they can survive, then when we eat, we get more nutrients. So about survival, it must survive the environment, so drought resistant, then it must survive or prevent itself from getting eaten by animals so that we can eat them. So these two survival, survive environment, prevent itself from getting eaten. Then if they have higher nutrition value for us, it is like cherry on top. So it is not difficult to remember the points on better variety. Then the last thing is C. C is for cheaper. So medicine produced in such a way, such as insulin, is lower in cost, cheaper, and is a more effective methods of medicine production. Then you might see the bracket, no complaint from vegan. Then you might be wondering, hey, why vegan complain about this? Or complain about other method? Because it's more effective because the insulin produced is human insulin. Before genetic engineering, we actually collect insulin from animal, for example, pig. So vegan will complain that, oh, the insulin actually come from animal. Don't kill the animal, don't harm the animal. So vegan don't like, vegan will complain. So it's not very effective to get from animal. So with genetic engineering, we can get a hell lot of insulin from just bacteria that have the human insulin gene, which is expressing itself, producing insulin. With benefits will also comes its problem. So here are some of the cons. First, let's talk about the social and the health issue. Because the plant is producing a new protein, it might cause allergies to some people. And those proteins might get involved in reaction which will cause them to become toxic. Some of them are toxic themselves. Or those proteins might get involved in reaction causing them to become carcinogen. Carcinogen is just a fancy way of saying agent which causes cancer. So those proteins might get metabolized into some cancer-causing chemicals. The next point is all about gaining or developing resistance. Bacteria that causes disease might gain antibiotic resistance. Pests might develop toxic resistance because only those that doesn't get killed by the toxic will survive. So in the end, the future generation of those pests will have the resistance to the toxic. Then weeds which competes with the crops might gain the pest-resistant gene from the crops itself. Then weeds which competes with the crops which we are growing might gain the pest-resistant genes from the crops itself. And because it is possible to gain all of this resistance, some country might go and develop a virus or bacteria that have tons of resistance and use it to infect people. So this technique might be used for chemical or biological warfare. Then some of you might want to ask the important question of how to memorize all of this. We can look at some of the words such as allergy, start with A, toxic, start with T, gaining, start with G, chemical, start with C. You have ATGC. Doesn't it sound familiar like something that we have learned in the previous lesson? Like the ATCG which we learned. So when it comes to social issue, just remember ATCG or ATGC, up to you. Allergy, toxic, gaining of resistance, and chemical or biological warfare. Because DNA is part of us is related to social issue. So this is how you remember the 4 points. Allergy, toxic, gaining of resistance, and chemical warfare. A, T, G, C. As for ethical issue, so the difference between ethical issue and social issue is social issue will affect more people. Ethical issue might not even affect people. So what does ethical issue affect? First, animal might suffer because when we are doing experiment, we might breed animal just for research purpose. And it can be very painful for them because we are just giving them drugs or testing them with chemical to see whether it is toxic to them. So those animals might just be bred to suffer. Pest-resistant crops can kill useful insects such as bees because pest-resistant crops will produce toxic. Another issue is only the rich can afford such technology. One example is the gene therapy, which replaces a faulty gene with a working one so that the person will not get a disease that he is supposed to get. And in cases whereby animal genes are inserted into plant, vegan might object consumption of plant containing animal genes. So vegan again. And again, the important question, how to memorize this? So ethical issue, when we talk about ethical issue, people like all high up in the air. So we remember air, animal, insect, and rich, AIR. So animal bred for research purpose will suffer. So it's not ethical to just breed animal where their whole life is suffering from toxic, suffering from cancer. I for insect, useful insect might die. R for rich, only sugar daddy can afford this technology. Then the last part on vegan, I think you just remember lah, because it is funny as. Lastly, let's compare the difference between selective breeding, which we learned in the previous chapter, and genetic engineering, because some of you might get confused, since both of it will resulting in offspring that are better. So let's look at selective breeding and genetic engineering and compare them. For selective breeding, we need to pair up organisms of the same species so that they can reproduce, then we can select from the offspring the quality which we want. For genetic engineering, any gene can be inserted into any organism. Animal gene can be inserted into plant, which is why vegans are complaining. During selective breeding, undesired gene might be passed on. For genetic engineering, only genes which are selected are being passed on. And because for selective breeding, undesirable genes are being passed on, it is a relatively slow process and less efficient, since you might take a few steps forward, then a few steps back. So it requires many generations. For genetic engineering, it is quick and more efficient, since the specific genes are being passed on. So normally it requires just 1 or 2 generations. For example, if you just want to insert one gene, you just need 1 generation. If you want to insert 2 genes, then maybe 2 generations. And for selective breeding, because we need a lot of the animal or plant so that we can select those with the desirable traits that we want, we need a large amount of space. For genetic engineering, we can just do it in a lab. So the space required is a lot less than the space needed for selective breeding. And that will be all for the chapter of molecular genetics.